Damaged mitochondria are usually considered a sign of aging, but scientists have repeatedly found a strange exception. In some animals, slightly disrupting mitochondria can actually make them live longer. The damage itself is not the secret. A new study published on August 13, 2026 suggests that survival depends on whether the cell can recognize the damaged mitochondria, physically separate them from the healthy network, and remove them before the problem spreads. And surprisingly, the signal controlling this process does not begin inside the mitochondria. It comes from another cellular structure called the endoplasmic reticulum. Researchers found that calcium released from the endoplasmic reticulum activates a chain involving the cell's internal skeleton. That machinery reshapes the mitochondrial network so damaged sections can be isolated and cleared away. When the researchers disabled this response, the same mitochondrial stress that normally extended lifespan stopped doing so. The discovery helps answer a major paradox in aging biology. Why can mitochondrial damage sometimes shorten life, yet under other conditions make an animal live longer? Mitochondria are often described as the power plants of the cell. That comparison is useful but incomplete. They convert nutrients into usable cellular energy, but they also help control metabolism, calcium, stress responses, inflammation, and cell death. And mitochondria are not usually sitting inside cells as thousands of completely separate beings. They constantly join together and split apart. At one moment, several mitochondria may fuse into a connected network. Later, part of that network can divide. This flexibility allows cells to redistribute resources and respond to stress. It also creates a quality control system. Imagine one section of a city's electrical grid begins malfunctioning. You would not necessarily destroy the entire grid. You would isolate the damaged section, disconnect it, repair or remove it, and keep the rest running. Cells appear to do something similar with mitochondria. The new study used the tiny roundworm Caenorhabditis elegans, one of the most important experimental animals in aging research. The researchers studied worms carrying a mutation in a gene called NUO6. NUO6 is part of mitochondrial complex 1, one of the major protein complexes involved in energy production. Disrupting complex 1 sounds like something that should be harmful, and mitochondrial complex 1 dysfunction can absolutely cause disease. But this particular form of mitochondrial stress in C. elegans produces an adaptive response associated with longer lifespan that creates a useful experimental model. Instead of simply asking what happens when mitochondria fail, scientists can ask what determines whether the cell adapts successfully. The researchers focused on a calcium channel called imply. It sits in the membrane of the endoplasmic reticulum, or ER. The ER performs many jobs, including protein processing, lipid production, and calcium storage. Calcium is not simply a mineral used for bones. Inside cells, calcium behaves like a rapid communication signal. A small pulse of calcium released in the right place can activate proteins, alter metabolism, and change cell behavior. The researchers found that the longevity produced by the NUO6 mitochondrial mutation depended on NC-free air. When this calcium signaling pathway was disrupted, the lifespan advantage disappeared. At first, the obvious explanation seemed to involve energy production. Calcium can enter mitochondria and stimulate enzymes involved in respiration. Perhaps calcium was simply helping stressed mitochondria produce more energy. The researchers tested that possibility. They disabled the mitochondrial calcium uniporter, the major pathway used to move calcium into mitochondria. But mitochondrial calcium uptake through that pathway was not required for the longevity effect. The story had to be happening somewhere else. Then the researchers looked directly at mitochondrial structure. In worms carrying the longevity-associated complex 1 mutation, mitochondrial networks expanded. That may initially be useful. If individual mitochondria become less efficient, the cell can compensate by building more mitochondrial material. But compensation creates another problem. Damaged mitochondria now need to be separated from that larger network. When imply signaling was impaired, the expansion became excessive. Instead of a controlled adaptive network, the worms accumulated large abnormal mitochondrial structures. It was as though the cell knew it needed more power plants but had lost the ability to dismantle the broken ones. This pointed researchers toward the cytoskeleton. Cells contain a dynamic framework made partly from a protein called actin. Actin is famous for helping muscles contract, but inside nearly every cell it also forms temporary structures that move components, change cell shape, and organize internal machinery. During mitochondrial stress, the researchers observed a dramatic increase in actin structures. The response depended on imply calcium signaling. Then they interfered with ARP2/3, a protein complex that helps build branching actin networks. The stress-induced actin remodeling disappeared, and something else happened. Mitochondrial quality control failed. Damaged mitochondrial networks expanded abnormally, similar to what happened when imply signaling was disrupted. The lifespan benefit was also suppressed. Now the pieces were beginning to connect. Mitochondrial stress triggers calcium signaling. Calcium helps activate actin remodeling. The actin network helps reorganize mitochondria. Damaged sections become separated. Then the cell can dispose of them. This last step involves autophagy. Autophagy literally refers to cells breaking down and recycling their own components. It is not random self-destruction. Cells can package damaged material and deliver it to lysosomal systems for degradation. When mitochondria are selectively removed through this process, it is often called mitophagy. The researchers found that interfering with autophagy produced the same general problem. Mitochondria accumulated, and the longevity advantage disappeared. This suggests that building new mitochondria is only half of a successful stress response. The cell must also remove the bad ones. Think of maintaining a fleet of vehicles. If aging trucks begin failing, buying more trucks can temporarily maintain delivery capacity. But if you never remove the broken vehicles, the parking lot eventually fills with machines that consume space and maintenance resources without doing useful work. Longevity may depend on balancing creation with destruction. The researchers tested this idea one more way. Instead of restoring calcium signaling or actin remodeling, they directly pushed mitochondria toward greater fragmentation. Breaking the giant mitochondrial networks into smaller units improved the abnormal mitochondrial expansion caused by defective imply signaling. More importantly, it rescued the lifespan phenotype. That is powerful evidence for the mechanism. The problem was not simply low calcium. The downstream physical organization of mitochondria mattered. But this study does not mean mitochondrial damage is healthy. That would be one of the easiest and most dangerous ways to misunderstand the research. Severe mitochondrial dysfunction causes real human diseases. Mitochondrial damage is also associated with neurodegeneration, muscle weakness, metabolic disorders, and aging. The study shows that one specific type of mitochondrial stress in one experimental organism can activate a protective response. The benefit comes from successful adaptation. Stress without sufficient adaptation can be harmful. This idea is sometimes called hormesis. A small challenge can activate defenses that leave the organism more resistant afterward. Exercise is a familiar example. A workout temporarily stresses muscle, energy stores fall, proteins are damaged, reactive molecules increase. Yet during recovery, the body adapts. The result can be stronger muscle and greater metabolic capacity. But increasing the stress indefinitely does not produce infinite benefit. At some point, the damage overwhelms the ability to recover. Mitochondrial stress appears to follow the same principle. The new research suggests that calcium signaling and mitochondrial cleanup may help determine which side of that line a cell lands on. There is another reason the finding is interesting. It shows that aging mechanisms cannot always be understood by studying one organelle at a time. The mitochondria were stressed, but the crucial response required the endoplasmic reticulum. The ER communicated through calcium. Calcium changed the actin cytoskeleton. Actin altered mitochondrial structure. Autophagy removed the damaged material. Several cellular systems had to cooperate. Aging may therefore arise partly when communication between these systems begins breaking down. Could scientists eventually use this mechanism to slow human aging? That is completely unknown. The experiments were performed in C. elegans. A worm has only about 1,000 somatic cells and lives for weeks, not decades. Its mitochondrial stress responses share many components with mammals, but that does not mean manipulating INSP3 actin or mitochondrial fragmentation would extend human lifespan. These pathways are also essential for normal biology. Calcium signaling controls heartbeat, muscle contraction, neurons, and many other processes. Actin is used throughout the body. Autophagy must be carefully regulated, and deliberately fragmenting mitochondria could be harmful depending on the tissue and disease. There is currently no treatment based on this study that has been shown to slow human aging. No supplement has been demonstrated to reproduce the effect, and the study provides no reason to intentionally damage mitochondria. The value of the research is more fundamental. For years, scientists have known that some mitochondrial mutations extend lifespan while others shorten it. Now they are beginning to understand why. The crucial variable may not simply be how damaged the mitochondria become. It may be whether the cell can remodel itself fast enough to contain that damage. Healthy mitochondria are not necessarily mitochondria that never experience stress. They may be mitochondria living inside a system capable of recognizing failure, separating damaged components, and recycling them before they contaminate the rest of the network. That changes the image of healthy aging. A young cell is not a perfectly preserved machine. It is a machine that constantly repairs itself. It detects problems, it reorganizes, it removes what no longer works. And then it continues. The worms in this experiment lived longer not because their mitochondria were perfect. Their mitochondria were deliberately impaired. They lived longer because the rest of the cell responded correctly. The lesson may extend far beyond mitochondria. Aging is filled with damage. The question is whether damage accumulates faster than repair. And this study suggests that one of longevity's most important skills may simply be knowing what to throw away.